Back in chapter 5, we learned that we could use pedigrees as a genetic family tree in order to establish the inheritance patterns and the genotypes of a particular trait within a family. Now, we need to do this because humans are not model organisms. We have a small number of offspring. It takes a long time for us to mature and to have children and to grow old. And so sometimes it takes a long time to study human traits. And so we can use pedigrees as sort of a way to look back and discern the inheritance pattern. So just a quick review, if we have an autosomal dominant trait, these tend to appear in every generation once they pop up in a pedigree. And the important thing here is that if you have affected parents, they can produce both affected and unaffected children. And this is because they can be hiding that recessive in the parents, but still pass it on to the children. But in order for that to happen, they have to be heterozygous. If the trait is autosomal recessive, this is the one that tends to skip a generation, so to speak. Unaffected parents can produce an affected child. So this means that the parents also must be heterozygous for this to happen, and they have to pass on both of their recessive alleles to the parents. So we can also use these same ideas for X-linked dominant and X-linked recessive traits. Now, the example that I used previously with color blindness and the white eye color are both X-linked recessive. And this is going to be the most common one that we see because they tend to be a bit more common as far as disorders are concerned. Now, the dominance pattern works the same way in that you can have two unaffected parents producing an affected child. But the difference here is that you have to look at the ratios of males to females. So if you look at this pedigree, we see that the majority of individuals in this pedigree that are affected are males. So when there are more males affected than females, or many more than you would expect, you might think that this trait is an X-linked recessive. Now, keep in mind, females can have this trait. And so the fact that a female has the trait doesn't make it not X-linked recessive. But when you have a lot of males compared to very few females, you might think that it is X-linked recessive. Now, if this was normal X-linked recessive, if it was normal recessive, autosomal recessive, you would expect roughly 50-50 males and females. You might not have exactly, but you would have roughly the same amount of males as females. But the key thing with X-linked recessive is that mothers pass it on to their sons. Remember that crisscross inheritance pattern. And fathers pass it on to their daughters. Males cannot ever pass it on to their sons. So that's another key. If you see a male passing it to a male, it's not X-linked recessive. X-linked dominant is a little bit harder to determine. In X-linked dominant, males pass, again, only to daughters, but females can pass to both. And this is, again, sort of hard to see sometimes. But if you look, you can see we have kind of the opposite pattern that we have on the right there. Fathers pass to daughters instead of mothers passing to sons. The key thing with these X-linked traits is that you can't 100% confirm that they are X-linked based on the inheritance pattern. And this is just due to the fact that you can have both sexes with the trait. And so really to confirm an X-linked gene, you have to map it to the chromosome. You have to sequence it, or you have to do mapping, which we have not done yet. But we've got some of our conditions down here. X-linked dominant, 100% incidence of affected daughters from affected fathers. And for X-linked recessive, 100% incidence of affected son from affected mother. Now remember that with X-linked recessive, it's less common in females, so you often don't have affected mothers. So you have to look at several different things. So let's look at a couple of different pedigrees. This is the Hemophilia Pedigree for Queen Victoria. I mentioned that at the end of the last video. Queen Victoria is thought to have been a carrier. So carriers are denoted with yellow circles here. because her father had hemophilia. And then again, because there was a lot of inbreeding within the European royal families, you can see one here. These guys are actually cousins. You see sort of a concentration of X-linked disorder or X-linked characteristics within this population. So if you look at this, there are actually no affected females in this pedigree. This is quite a large pedigree, 4 generations with a lot of individuals. The only thing we see are carrier females. All of the individuals who are affected are males. So down here in this generation, we have a lot of males that are affected. And then, of course, we could fill in all of these genotypes if we wanted. If we wanted to say that H was normal and lowercase h was hemophilia, we could start with Queen Victoria. So Queen Victoria is a carrier because her son Leopold had it. So she would be heterozygous on the X chromosome. Her husband didn't have it. And because he only has one X chromosome, we know that it is a dominant allele. So all of these females are going to be heterozygous, and all of these males that are affected are going to be hemizygous for the trait. Now we could go through and fill out a lot of them. We are only going to focus on one small portion here. So this is, let me go back. This is the center portion here. This is Anastasia and Alexis Romanov. So this is the Russian Romanov. This is a Russian royal family in the early 1800s. So Anastasia and Alexis are both thought to have been killed during the revolution in Russia. So we don't know what Anastasia's children would have been, but we can kind of think about what she would have been. Let's fill in some genotypes. Her mother is a carrier, and her brother is affected, so we know that her mother has to be a carrier. Her father is unaffected, and she is unaffected. But let's say that we know that she is a carrier. She actually may or may not be a carrier. Oops, excuse me. I got a typo there. But let's assume that she is, in fact, a carrier. So she could be normal. But let's assume that Anastasia is a carrier. So let's say that she is heterozygous. Now, if she marries within the royal family, there's a decent chance that her husband could have hemophilia, although this does tend to be tends to cause early death. So it's probably pretty unlikely that she would marry somebody that also has hemophilia. If she did marry a husband, marry a man and have children with hemophilia, then we could get the possibility where some of her female offspring could have the disorder and some of her male offspring could have the disorder. Let's say that she does not marry somebody with hemophilia, which is probably more likely. In this situation, we are going to have normal females, carrier females again. So carrier females in this family tree are pretty common. Normal male and colorblind male. So again, we have the 1 to 1 to 1 to 1. And again, this is 1 normal female to 1 carrier female. So I'm writing carrier because we're specifically looking at carriers here. Normally, if it's phenotype, you would just say she's normal. 1 normal male. Oops, I'm sorry. I'm having trouble with males and females. And 1 hemophiliac male. So 1 affected male. So let's look at another couple of pedigrees. This one on the left also shows carriers. So again, the key thing here is that in this case, all of the affected individuals are males except for one female. So this is going to indicate that it is an X-linked trait. And then to determine recessiveness, we're going to look at parents. So here we have two sets of unaffected parents. The females are carriers, but they are unaffected, and they produce affected males. So this is going to suggest that it is recessive. So we have an X-linked recessive here, and we can see that a little bit easier in this pedigree on the right. We have unaffected parents with affected children in every generation. So this is a good example of skipping a generation. But we also see that it occurs, in this case, is only males in this pedigree. So again, we can see that it is X-linked recessive. Last one, this is an X-linked dominant. So we can look for indications of dominance. We have an affected parent. They always produce affected children. That is a strong indication that this is a dominant trait. Now, we can't tell for sure. This is not really 50-50 here. I'm sorry, it's closer to 50-50 than it is to more males than females. So this is going to tell us that it is not X-linked recessive. But we can look and we see that fathers pass to all daughters. So when fathers pass to all daughters, there's no apostrophe there. Fathers pass to all daughters. That's a strong indication that it is probably an X-linked dominant. Over here, we've got individual 3-8. 3-8 is a female with the disease. I'm not sure what's going on there. She has passed it to both father and, she didn't pass it to her father, both male and female children. I do know what's happening there. It's a dominant trait, so she's getting it from her mother. The key here, though, is finding this situation where fathers pass to all daughters. And we see that, again, in this generation here. There's only one daughter to pass it to, but he does pass it to her. All right, so we have sex-linked traits. These are traits where the gene for the trait is actually found physically on the X chromosome, and can be Y as well, but in general, we're going to be talking about X-linked genes. We can also have sex-influenced inheritance. So this is inheritance where the phenotype is affected by the sex of the individual, but is not located on the X chromosome. So this is autosomal inheritance, so a trait that is on one of the autosomes, but it is actually affected by the sex or the hormones of the individual. And I see I actually have a typo. I will fix this in your printed notes. This should say sex-limited. Sex-limited and sex-influenced inheritance. So these are two different types of inheritance where the genes are autosomally encoded but affected by the sex of the individual. So let's look at a couple examples of these. So the first one, the one I like to use, and the sort of the most obvious one I think for students to see, is sex-limited inheritance. So this is where a trait is encoded on an autosome, so not the X or Y, but that trait is expressed in only one sex. So all individuals will get the genes, but it's only expressed in one of the sexes. So both sexes have the genes, but only one displays the phenotype. And this is because the hormones of the individual are affecting development, or something else is affecting development, and so that gene might not be needed. So a good example of this is milk production in mammals. So I've got a cattle here as a specific example. I really like these cows. They make me laugh every time I see this slide. So all cattle have genes to produce milk. But only females do because they have to feed their babies. So this makes sense, right? Females have mammary glands. Males also have breasts, but they are, and males don't have udders in cattle, but in humans, males also have breasts, but they don't produce milk, just like male cattle do not produce milk. Now, this is related to the fact that there is female development, production of udders, production of all of the sort of mechanical parts that produce the milk. So even though all cattle, male and female, have genes that would allow them to produce milk, only females actually produce milk. And of course, you know that is related to hormones. So another example of this sex-limited inheritance is cock and hen feathers. So this is related to the types of feathers that are found on male and female roosters. So if we look at this male rooster here, we can see that his feathers are much more pronounced. They're flashier, they're longer, they're more curved, and on this hen they are shorter, not quite as prominent, not quite as showy. So they're less curved, they're a little bit smaller. So individuals that are heterozygous or homozygous are going to be hen feathered, whereas individuals who are homozygous recessive are going to be cock feathered. Now, even if a female has the homozygous recessive allele complement, she is not going to be cock feathered. So no matter what her actual genotype is, she's always going to be hen feathered. So these guys over here, these are called Seabright bantams. All of these individuals are HH. So there's actually no dimorphism. They're all hen feathered. Over here, we've got a leghorn rooster, like foghorn leghorn up there on the left. Again, these guys are always going to be homozygous recessive, but the females still show hen feathering. So here we have the sexual dimorphism no matter what the genotype is. So we are limiting our traits to one sex or the other. The other is a sex-influenced pattern of inheritance. This means that it can be expressed in both sexes, but it's usually more common or more strong in one or the other. So an example here is pattern baldness. So you can see here this poor man has lost most of his hair. This woman has very thin hair, so she's not actually bald, but she has less hair on top. So again, this goes back to those autosomal genes that are affected by the hormones of the individual. Now we see this pattern baldness most strongly in heterozygous individuals. So in these guys, heterozygous individuals express one phenotype if they are male and one phenotype if they are female. So for this trait, all individuals who are homozygous for dominant are going to be bald, or in the case of females, have thinner hair, regardless of sex. So females have less pronounced baldness, males are going to be bald. Individuals who are homozygous recessive are not going to be bald. So no baldness. I guess that just means they have hair. They will not be bald. But in the heterozygous individuals, females are not bald. Males are bald. So it really just depends on the sex of the individual. And again, this relates to hormones. So we can see that in the homozygous dominant genotype, we get bald or we get less hair if we're female. So you can see that even though individuals might have the same genotype, depending on the sex, they're going to have a different phenotype. Now, it's not quite as strong as, say, our hen feathering or our cock feathering, but in this case, we do have a difference again between males and females. All right, so let's talk about sexual differentiation in humans. So this all goes back to the Y chromosome. The Y chromosome is going to contain the genetic material that is necessary for producing what our book calls maleness. So characteristics that are unique to males. So at around 7 weeks of gestation, this is 7 weeks after fertilization of the eggs, the gonad cells, these are the cells that are going to develop into either the ovaries or the testes. Gonad cells in an individual that is X and Y are going to develop into the testes. So these are the cells that are eventually going to produce sperm. If there is no Y chromosome present, gonads do not develop into testes, but instead they develop into ovarian tissue. So if those genes are not there to direct the development of the testes, the individual is going to develop ovary tissue. And then after the gonads sort of pick their track based on XY or XX, you get the development of other tissues associated with the testes and the ovaries. So you start to get the sex organs developed and things like that. So let's talk about this Y chromosome and why it is so important in male development. So you might recall from previous karyotype images that X chromosome is quite large. The Y chromosome is quite small. The Y chromosome only contains about 75 genes compared to the X chromosome that contains over 1,000 genes. So the difference here is that almost all of these genes on the Y chromosome code for male development. As I said earlier, some of those genes are homologous. Most of them are not. Really, they are regions that are sort of homologous with the X, sequence regions. And the reason for those sequence regions is so that the X and Y can pair during meiosis. Those regions that are homologous is called the PAR region, the pseudo-autosomal region. So this is just saying that this region sort of behaves like it's an autosome. It has homologous structures to the X chromosome, which allows them to line up during meiosis. And then we have the important part. We have the MSY region, the male-specific region of Y. And this is the region here that is going to be specific to male. So it's not found in the X chromosome. Actually, it's this whole region here. But not all of it contains functional genes. We've got some heterochromatin here that doesn't contain any genes. The really important part is this SRY gene region. This is the sex determining region of Y. So these are going to be the important genes that are involved in male development. So about 95% of this Y chromosome is not homologous with the X chromosome, but it is still important in lining up with the X chromosome, and there is a little bit of crossover exchange between them because remember, crossovers have to occur in meiosis so that everything separates properly. So as I said, that SRY region is really important in male development, and that's because it triggers the development of testes in mammals. So we've got a little diagram here that shows the difference between males and females. I already talked about this a little bit, but basically this SRY gene produces something called the testes determining factor. So this is a protein that is going to, it's a transcription factor that is going to affect the genes that are needed for testes differentiation. So this SRY region produces a protein that is going to turn on expression of all of those other male-specific characteristics. So if you have a Y chromosome, you're going to produce testes. Now we're going to talk about that XXY situation in just a minute. And then of course testes will eventually produce sperm and continued male sexual development of organs. Without the SRY gene, so that gene's not there, we do not get that testes determining factor. So those genes that are needed to cause development and growth of testes aren't there. And so instead, those gonad cells are going to develop into ovaries and additional female characteristics. So you can see here we have an absence of that Y chromosome. So we're going to talk about these individuals, these XXY and XO individuals, in just a minute. The important thing now, though, is to just remember that the presence of the Y chromosome is going to induce male sexual development, and the lack of the Y chromosome is going to induce female development. So the source of those strange sex chromosome complements is actually nondisjunction. So we saw nondisjunction in our last chapter when we talked about Down syndrome. Nondisjunction comes from an error in meiosis where your chromosomes don't separate properly. So usually this comes from a lack of microtubule formation so that your chromosomes are not pulled apart correctly. So I have a question here for you. In which gamete, the sperm or the egg, does the XYY condition occur? So this is a different one. So this question is asking, is a male or a female going to produce this XXY condition? And then where does it happen? In which meiotic stage does it happen? So just stepping back and thinking critically about this, we sort of already can answer the first question. It has to occur in the sperm because of the Y. Y is only found in the sperm. So it has to occur in the process that is going to produce the sperm. So let's draw out meiosis 1 and see what happens. So if we are going to produce a sperm, we're going to start with male cell in the gonads, and in the S phase of the cell cycle, we're actually going to duplicate our chromosomes. Now let's say that meiosis 1 occurs. So we're going to get 2 cells here. Now let's say that in meiosis 1, nondisjunction occurs, and so our homologs, which is what we separate in meiosis 1, are not separated. So that means that our chromosomes are not going to pull apart. So this cell down here is going to get no sex chromosomes. So then if we just continue this one, we don't have to follow this line because now we have no sex chromosomes. So we can't have a condition where we have extra because we have none here. So let's say now that we go through meiosis 2 and we separate our sister chromatids. Remember, sister chromatids are the identical copies. So if we separate our sister chromatids, now each one of our cells is going to get an X and a Y. So this individual grows up. Now, since this is a sperm, we need an egg. Eggs are always going to have an X chromosome. So if this egg fertilized one of this individual's sperm, they are actually going to be XXY, which is not the condition that we need. So we know that the non-disjunction has not occurred in meiosis 1. So let's see if we can get it in meiosis 2. So here, again, we are going to start with male chromosome. So we have, male cells. So we have in the testes our X and Y cell. We're going to go through meiosis 1. Again, actually this is going to be S phase. We're going to duplicate our chromosome. Meiosis 1 is going to occur normally, and we are going to separate homologous chromosomes. So each of these cells is going to get, one's going to get a set of X's and the other is going to get a set of Y. So now let's follow this one here. We'll go through meiosis 2, where we're going to separate sister chromatids, and we're going to get 2 cells here, both with an X. So this is a normal situation. We're going to get a cell with normal X chromosome complement. The other option here would be for the non-disjunction to occur and to end up with an XX and an O, which is what we found in the previous one. This is, is also not going to give us XYY for the same reason that the previous slide didn't give us XYY. But now let's go through this other cell, the Y chromosome. Let's say that non-disjunction occurs there and the Ys don't separate, our sister chromatids don't pull apart, and so we're left with a cell with 2 Y chromosomes and then another with no chromosomes. So this is also not going to give us the right situation, because if this cell is fertilized by an egg, we're going to end up with XO, which we'll talk about in just a minute. If this one is fertilized with an egg, we are going to end up with our XYY. So this tells us that this XYY condition occurs in males, because we need the Y, in meiosis 2. So that question was really just a thought experiment. Let's talk about a couple of specific syndromes that actually helped us to understand the importance of the X and Y chromosomes in normal human sexual development. So in the 1940s, scientists identified 2 human chromosome abnormalities that are due to non-disjunction. In these abnormalities, we have an incorrect number of sex chromosomes. So in the first one, we have Klinefelters, which is an individual with 47 chromosomes. You might recognize this notation from previous lectures. 46 chromosomes, and then here it's showing that it has an extra X chromosome. So we have 2 X's and a Y. So this Y is going to give some male characteristics. Turner syndrome is actually the lack of the other sex chromosome. So I was writing this one as XO earlier, meaning no other chromosome. So they're lacking either the other X or the other Y. Now note that this one has a lack of Y, which means that these individuals are going to tend to develop to be a bit more, have more female characteristics. Again, the 45 indicates that we are missing a chromosome. So we've got the karyotypes here. Here's our XXY. This is our Klinefelters. And then our single Y, which is Turner. And then down here on the side, we have normal chromosomes, normal female. Here we have 2 X chromosomes. Now again, both of these are going to be due to non-disjunction. So let's look at Turner syndrome a little bit. So this is characterized by an extra X chromosome. And in this syndrome, remember, syndrome means a set of characteristics for a disease. Individuals tend to have some particular characteristics. So they tend to have longer than normal appendages. They have male genitalia because they do have that Y chromosome. But they don't have sperm production, and they don't have full development of the testes. They also tend to have some female characteristics, such as slight breast enlargement and then more rounded hips than would be normal in males. This also does affect mental intelligence. Individuals with Klinefelter syndrome tend to have below normal intelligence, and this occurs in about 1 in 660 male births. Female development is not fully suppressed in this syndrome, and so you do get some of those female characteristics. And this can lead to some social problems as well, because individuals tend to have sort of ambiguous sexual development. And so this can, of course, lead to some other social and emotional I guess I don't want to say problems, but social and emotional development problems. And then Turner syndrome in females. This is where a female is missing one of the X chromosomes. Because there's no Y chromosome, there are no testes development, and so development tends to be more feminine. There tends to be female external genitalia, but ovaries aren't well developed, and so individuals tend to be sterile. They tend to have smaller stature and underdeveloped breasts, but unlike Klinefelter's, this one doesn't seem to affect intelligence at all. And it's a little bit less common than Klinefelter syndrome, only about 1 in 2,000 female births. So just sort of a summary. I didn't have a summary slide in here, but I decided to make one. Klinefelter and Turner syndrome were just really important in understanding the importance of both sex chromosomes, so the function of the X and the Y. Particularly the Y chromosome. So the understanding that a lack of a Y chromosome leads to no male development. And so sort of by default, when there's no male development, there is female development. So these two syndromes really helped us to understand sort of the importance of having two sex chromosomes and having the proper complement of sex chromosomes.